Hello, and thank you for coming. I am Katharina Glomb. I'm a postdoc in Petra Ritter's group at the Berlin Institute of Health at the Charité, and I'm going to talk to you about harmonic modes and what they have to do with waves. But before I do that, I just want to thank the organizers very much for inviting me to give this talk, and I will do my best to tell you something that you don't know yet. Okay, let me share my screen. Here is. Okay, so let's get started. I will begin with a bit of a vague intro to what harmonic modes are, and that will hopefully show you why I find them so fascinating. So harmonic modes describe spatial patterns that appear everywhere where we have any kind of standing wave, be it mechanical, like here in the violin example, where we have vibrating strings. So of course you will primarily think about the tone that the string elicits, but think of the fact that also the string vibrates and has a certain spatial frequency. So this spatial pattern is a standing wave, which is linked to the tone that you hear. And another example for a mechanical vibration is something called Chladni plate. You can Google this, there's really cool YouTube videos. And I must admit that right now for the first time, I'm realizing that this plate is apparently also excited with a bow. So you can see here the violin bow, I think. And so it's used to make this plate vibrate. And the lines that you see are made up of very fine sand grains. And if you get the right tone, and it's the right tone in relation to the shape and the size of the plate, you will also get a standing wave pattern, and the sand will settle into the parts of the plate that stand still during this vibration. So beyond this kind of mechanical vibration, you also can have something like here, these animal codes, where you have rather something that is diffusing and it has to do with gene expression and reactants moving around in these biological domains, but also these code patterns can be explained by harmonic modes. So the idea that harmonic modes also play a role in the brain is not particularly new. Already in 1980, you have this paper from Swindale and others that shows that ocular dominance patterns in the macaque cortex can be explained by harmonic modes. And when you look at big global patterns of activity when using MEG or EEG, you can explain them, you can explain a lot of them using spherical harmonic modes. So also these standing wave patterns show up in EEG and MEG recordings. Going one step further, you can project back spherical harmonics onto the cortical surface. And despite this kind of hole caused by the corpus callosum between the two hemispheres, you will still see patterns that are very reminiscent of known functional and structural networks. And this was shown by Robinson and others in 2016. So the actual meaning of the word harmonic derives from simple harmonic motion. And since I'm not a physicist, I don't really know what I'm talking about. So I will cite Wikipedia when I tell you that a harmonic motion is a periodic motion or oscillation, which I've already mentioned, that arises because there's an object that is displaced in space, for example. And at the same time, there's a restoring force that pushes it back to its equilibrium position. So force and equilibrium are really important terms here. And you can imagine, for example, a spring that you pull down, and then because there's tension in this spring, it gets pulled back to its equilibrium position, and then it starts oscillating. And if there's no friction, this could go on forever, right? Later on, I will talk a bit about Fourier analysis, and I just marked this here so that you know that I'm not making this up, but that Wikipedia is behind me. So from this simple harmonic motion, where you may wonder, what does this have to do with the brain? We can go to more complicated motion using an analog of Fourier analysis. And I'll show you a bit later what that does have to do with the brain. Okay, so let's talk about these patterns that I've been showing you, like the sand on the cladding plates or the spots on animal coats. Let's start with something very simple, or seemingly simple, that is diffusion. Or the heat equation is the same. So diffusion is a process in space and time, which you can see from the fact that this function u is a function of space x and time t, and there are derivatives of both on opposite sides of this equation. And for time, we have the first derivative, but we're not going to talk about time. It's not the topic today. I'm going to focus on space. And for space, we have the second derivative. So this can also be written in another way using this delta, which we will come to in a little bit. And we have, so the second derivative in space describes the spatial pattern. So we have structure in space and in time. Going to something a little bit more complex, we go to the wave equation, which is more relevant to the examples that I've already shown. And we see, again, the second derivative in space. We see this function of space and time. And we see that in this case, we have the second derivative in time. So this is how it differs from this other equation. But again, we have this mysterious delta, which probably for a lot of you is not that mysterious. So this second derivative, instead of leading to just like a plain constant concentration or temperature, it leads to a sustained oscillation, illustrated here with this rope example. And so we are interested in the shapes that something like this rope can take. Okay, so if you want to know the equilibrium point of a system, you want to know what it looks like once it reaches a state where nothing changes anymore. Note that nothing changes anymore doesn't necessarily mean that really nothing is moving anymore. It could also mean that there's a sustained motion, like an oscillation, as mentioned before. So all we have to do is set this expression to 0. This is the same expression as here. Sorry for switching between u and f. And we set it to 0 and we solve for f. And then the solutions that come out of this, the functions that we find that solve this equation, are called harmonic functions. So that's what harmonic functions are. So this delta is called the Laplace operator, or Laplacian, as several of you probably know. And as it turns out, solutions to this equation right here are eigenvectors of the Laplacian. So I will show you what that looks like later for the brain. But as we can see in these two equations, the Laplacian is the sum of the second partial derivatives in each direction of space. So this is down here. This is just a different way of writing the expressions that we see here. And because space and time are on opposite sides of this equation, we can separate them and solve them independently of each other. And this is why now we can only look at space and find the spatial patterns that occur during this equilibrium, at this equilibrium point, let's say. So this second derivative is also called the divergence of the gradient. And if that doesn't really tell you anything, maybe an intuitive interpretation of that is that this this divergence of the gradient is a scalar value for each point, which tells you if this point is larger or smaller or equal to the average of its neighboring values. So if you look for an equilibrium, you are looking for a situation where each point is equal to the average of its neighbors. And consequently spoken in a very like hand wavy and sloppy way here, just to give you an intuition, in a situation where each point is equal to the average of its neighbor, that means that the points aren't really pulling on each other anymore. So the overall pattern doesn't change. So you have a concentration or temperature distribution that is flat, or you have a sustained oscillation, for example. Okay. So in the cases that I've shown you, and also when we talk about the brain, there's a distinction between the domain and the signal. And this may be a little bit confusing if you don't think in these terms, but the domain is just the space in which the signal lives, let's say. So in the examples that I've shown, diffusion and wave equation, the domain is like a regular continuous space. And I'm not going to talk about this in like rigorous mathematical terms. Let's just say that this is the space that we're used to. We're so used to it, in fact, that we don't really think about it. So I think it's easy to think about the position of the robe at each point in time. So this just happens in the space that we're used to. When we think about the brain, it's maybe a bit easier to make this distinction because the signal that we measure is the stuff that we're familiar with, like EEG, fMRI, MEG, whatever it is that you measure. And the domain is the underlying graph, at least in network neuroscience. Of course, there are other approaches. You can look at the brain as a continuous sheet, but in network neuroscience, and for the purpose of this talk and for the purpose of my personal work, we look at a graph. So we conceptualize the brain as a graph, which is of course a very different domain from continuous space, because we have these points, they are not continuous, they are discrete, and the graph is somewhat irregular. Okay, and what's important to mention again here is that this graph is not the brain surface or the volume as it appears in this picture. So points that are neighboring in this graph are not necessarily the points that are neighboring on the surface or in the brain volume, but the connections between these nodes and consequently the distances between these nodes are set by connectivity matrices, structural, functional, or otherwise, whatever you choose, and which makes sense in terms of brain dynamics. Of course, this graph is deeply related to spatial architecture of the actual brain, which you can see from the colors. Yeah, it's color-coded in terms of these functional regions. This is the Glasser parcellation, the HCP parcellation, you're probably familiar with it. So all the red points are kind of clustered together, all the green points are kind of clustered together, but you know, this isn't a given, and it doesn't have like a brain shape. Okay, so why we do this? Because we know that the brain is a dynamical system, and the goal of all this stuff that I'm describing today is that we're trying to uncover the rules of the system. Because we don't really have a very good idea of the domain that the brain signal really lives in functional terms. All we have are some sample points. So our signal is kind of us sampling the dynamical system brain. And it's of course a very complex, high-dimensional system. And we want to understand this dynamical system. And all we can do is sample from it and hope that we are sampling the true underlying structure in a way that will help us reveal this structure. Another name for this underlying structure, which you've maybe heard, is manifold. This actual manifold is hidden from us. We're looking for it. Okay, and we can use this idea of the Laplacian to estimate this manifold, to get an idea of what this manifold looks like. Because remember, this Laplacian is the second derivative in all directions of space. So by looking at it, we can get an idea of what this space looks like and how it changes. And again, space and neighboring points is defined in terms of this graph. So how does the signal change? How does the graph change over space? Space defined by the graph. Okay. So okay, in the brain, the points correspond to brain regions or vertices, and the connections are set based on a connectivity, as mentioned before. And in the examples that I'm going to show, I'm going to focus on the case where we just use a binary decision of who's a neighbor and who isn't. Like, know that there are different ways of doing this, and I'm going to be a little bit imprecise here. I'm going to take some liberties here, but yeah, in this illustration, for example, you just see that there are connections between some nodes, but not between others. So it's a binary decision. Okay. If this all sounds very complicated, I can tell you that it's quite easy to do in practice. So here I just stole this figure from an excellent intro tutorial to an algorithm that is related to the one that I use. It's the UMAP algorithm. It's really easy to use. It's great software. I recommend also reading this tutorial if you just want to use similar techniques, because it's really very good. And in this toy example, they're just using a sine wave as an underlying structure. Of course, in the brain, the underlying manifold will not be something as simple as that. It will definitely not be one-dimensional, but you can imagine this is just to illustrate a point. So here you see that we have sampled points on the sine wave. And even though we don't know that it was a sine wave, the way that we've sampled the points and the way that we've connected the points based on who is next to whom, you see this underlying structure emerge, and we can estimate it, okay? So we can say that the graph is an approximation of the underlying domain for manifold. So here they are points on the sine wave. In the brain, the points are brain regions. Okay, it all sounds very complicated, but I can assure you that in practice, it's quite easy to do. So that's maybe a relief to hear that. It's easy to do. You would just have your passellation, you build your graph. The connections can be structural, functional, effective connectivity. And then you compute the Laplacian, for example, by extracting the binary nearest neighbor matrix. So you just take the strongest connections and you set them to 1 and you leave everything else out, and you do this up to a certain number of neighbors. And then you subtract this binary connectivity matrix from this matrix D, which is just a diagonal matrix. So it's 0 everywhere except on the diagonal where it has the number of nearest neighbors of each node. And then only one more step to get the harmonic modes. You perform an eigenvalue decomposition on this symmetrical matrix. And voila, you get eigenvectors and eigenvalues, and yeah, as you're used to. They are ordered just like always with eigenvectors, and I'm going to talk about the eigenvalues a bit later. But for now, just know that they are meaningful, and we're gonna talk about the order that they introduce in a bit. Okay. So by now, you're probably curious what these harmonic modes look like. So just to illustrate what they are, you've got some vectors now, and so you decomposed an n by n connectivity matrix where n is the number of brain regions or vertices or whatever you're using for your connectivity graph. So you can project the vector or like the values in this vector back onto your brain surface, let's say. And this is what I illustrated here, just for the first 4 eigenvectors. So here's the vector, then the same values are just projected back onto the brain surface. And here's also an illustration of what this looks like on the graph. And maybe you can get a vague idea of how this can be understood similarly to a standing wave, just like with this swinging rope that I mentioned earlier. You can already see how it's divided in different, the graph is divided in different clusters, let's say. Okay, so you have done an eigenvalue decomposition, which means that you get n eigenvectors as in any eigenvalue decomposition. So if you have n brain regions, you have n harmonic modes and you can play with them. Okay, now what are these? You see that even though we took a binary matrix, the result is continuous. So the magnitude of the entries in these vectors doesn't really have a meaning. They are eigenvectors, so they're normalized to length 1, and even the polarity is arbitrary. So you can, everything that's red here could be blue and the other way around. So the thing that matters is just the distance between the points as defined on this harmonic mode. So you can take any two brain regions in this vector and compute the difference between them. And this will tell you how different these two brain regions are in terms of their connectivity, depending on what kind of connectivity you used, of course. So this is how you read these colors. It's not directly like network membership. The more similar the colors, the more similar the connectivity pattern of the brain regions that you are comparing. Okay, so just to show you a few examples using the same data that I've been using for illustration, I'm showing you the first 3 harmonics and then another one that we found in analyzing some task data. So the first harmonic mode is depending on what method you use to build your graph and to build your Laplacian, is either constant or it's something like baseline activity, which is just the strength or the weight of each of the nodes. Then if you take the next one, you see this anterior to posterior connectivity gradient. I'm just going to drop this word here. So what this tells you is that frontal connectivity is very different from occipital connectivity, right? What the third harmonic mode tells you then is that the connectivity within the left hemisphere is very different from the connectivity in the right hemisphere. And here you can really see how this is a result of using structural connectivity. As you know, when you use fiber tracking, very often the long fibers between hemispheres are missing. So this is why this appears very asymmetrical. If you were to use functional connectivity, you would not see this division between the generalized hemispheres because the brain regions on opposite sides of the brain usually are very functionally related. Okay. And then this 10th harmonic is something that we found in an experiment. So people were looking at faces, we recorded EEG, and then we projected the EEG into the brain and analyzed the brain activity in terms of harmonic modes. And one of the harmonic modes that we found that activated strongly when people were looking at faces and processing faces was this harmonic number 10. And you see how it involves this fusiform face area, which makes perfect sense and tells you that these kind of functional connectivity patterns, these connectivity patterns that we found have something to do with brain function. Okay, so as I already said, what you find will depend on the connectivity that you used for your graph. And to make this a little bit clearer, here is an example from using functional connectivity. So in this case, the nearest neighbors are defined in terms of who has the highest correlations and not who has the most fibers between them, as before in the example. This one has a much higher spatial resolution. So this is why this pattern, these patterns appear much smoother and look kind of nicer. But never mind, the idea is the same. So what matters is that because this is function, you can see this nested hierarchy. And as I mentioned, you don't see much asymmetry, except here. Well, I'll talk about this. So we consider this hand area in the brain. Here you can see it much better. But so in this harmonic mode, in this functional harmonic mode, you see how this hand area is integrated into a large somatosensory motor network, which tells you that this entire network is highly interconnected and they have similar connectivity to the rest of the brain. If you go to a different functional harmonic, you see that the hand areas have different connectivity from other somatotopic areas. So this is an area that is related to the face, so your own face, so movement of the face and touch on the face. And this is an area, these are areas related to the two hands. So you see how the connectivity is very different from the point of view of this harmonic modes. So here they are segregated from other parts of the somatosensory motor cortex. And if you go to yet another functional harmonic, you see that the two hands are also different, which makes sense because you have two hands and they can do different things. So this is a beautiful example that shows how harmonic modes show structure on different levels, because it doesn't force each brain area to be part of exactly one network. And because I like this property very much, I'm showing you another example. And this one is about area MT, middle temporal area, or D5. This is what this arrow points to. And if you just look at the seed correlation of this area in the first row, again, this is using resting state fMRI, you see this pattern. So this is just like a correlation pattern that involves a big part of the cortex. But if you look at harmonic modes, again, functional harmonic modes, you can see that MT is involved in different networks with different functions. So in this mode, we see that its connectivity is similar to that of auditory cortex, involving also the temporoparietal junction, known to be involved in audiovisual integration. So this is a network that is responsible for audiovisual perception, basically. In this harmonic mode, we see the sensory motor pathway where we have visual areas connecting all the way up to the parietal cortex. And in this functional harmonic mode, you can see the ventral stream, including the face complex for recognizing faces. And interestingly, it also includes this face area that I mentioned before. So it's others, faces, and your own face. That has to do with this network. Okay, so the last point about interpretation that I would like to make is about gradients. I've already dropped this word accidentally earlier, but it's a topic that you may have heard mentioned. It's more and more popular, and harmonic modes play a big role in it, even though they're not always called this. And I mean, I'm not saying everyone should use the word harmonic modes, not at all. Gradients are a way of interpreting these modes that takes into account that the values in the vectors are continuous, as I've mentioned before. So you can start with a binary matrix, but you will still get continuous values in your spatial patterns. So if you just consider networks, kind of the way that I've interpreted it so far, you just, you cluster, you put them into chunks, and you don't really observe the fact that there's a continuous transition from one color to another, to one end of the gradient to another, right? So if you look at the continuum that these harmonic modes provide, you can interpret them as a smooth variation in connectivity pattern. So one great example of this, and probably the most well-known one, is this global cortical gradient that at this point has described many times, but I think the most well-known description is from this paper by Margulies and others from 2016. And it's a combination of two harmonic modes. We just reproduced the same thing. I'm just showing this to kind of underline how stable this finding is. So if we combine the first two harmonic modes and we plot them in their own space, so obviously the colors that I've been talking about in an illustrative way are linked to magnitudes, and you can just plot these magnitudes one against the other. And you can see that in one of the gradients, the somatosensory motor network and the visual network are close together, so their connectivity is similar in a sense, but in this other gradient, it's very dissimilar. And then kind of the rest of the cortex stands in opposition to that. And this is exactly what is shown here in this illustration, where you have visual areas in blue, somatosensory motor and auditory areas in green. And in red, you have higher cognitive areas, in particular, the default mode network, areas of the default mode network. So we have this triangle between somatosensory motor, you could say action, visual, you could say perception, and TMN, you could say cognitive processing. So this is, of course, a simplification, but you get the idea, and you can use the same interpretation for other modes, but this one is perhaps the clearest one. So it's not for nothing that we talk about processing streams and not processing jumps, right? Okay. Excuse me. So the last, what's going on with my slides? Okay, the last topic that I want to touch on, and this will be brief, is this relationship to the Fourier transform that I mentioned in the very beginning. So, and what might already be clear from the fact that harmonic modes are eigenvectors, that they form an orthonormal basis. So they are orthogonal to each other and they are normed. So what this means is that they can be used as a Fourier basis to express any pattern on the cortex as a superposition of the harmonic modes. This is illustrated here. I'm going back to the smaller, less smooth graph, but I've mentioned several times before that there's an equivalence between these patterns and the swinging rope with the different frequencies. So if you have a very simple domain, our usual domain, the circle or continuous line, then you have the sine and cosine waves, and you can decompose any pattern that lives on the same domain, which, as I said before, we don't usually think about the domain because we're so used to it, but any signal that lives on this domain, you can decompose into these waves, and then you end up with a spectrum. Now you have to make the flip to the spatial domain. So now we're talking again about the graph, but also here, any signal on this graph can be described as a superposition of the harmonic modes. Don't know why I put that. So in that way, harmonic modes are completely analog to sine and cosine waves. And this is not just, this is not just an illustration, but you can show this in rigorous mathematical terms, how it's exactly the same. So the difference only lies in the domain. The sine and cosine waves are ordered by frequency, and so are the harmonic modes. So don't be afraid of this slide. I will explain, just give me 1 second. So this is about the eigenvalues that I mentioned before. I already showed you that this example of hierarchical networks for the hand areas. And while that was, of course, a particularly nice example that I picked, it illustrates a general property of harmonic modes. And this is that they are ordered by frequency, just like sine and cosine waves in Fourier analysis. The frequency is a spatial one, and it's on the graph, so it's not that easy to see by eye, but the mathematical definition is clear, and I will show this to you. So if you first look at this figure again, maybe you can make out the general tendency for the chunks of cortex that have the same or similar color to get smaller as we progress to higher order harmonic modes. So first we have the full brain, then we have 2 halves, and then we have even smaller parts, right? So this is how we go from integration to more specialized brain areas, okay? Then here on the right, you can see the formal mathematical expression for that. And we start with the eigenvalue decomposition, and over here, which you should all be familiar with, so the eigenvalues are in this lambda, and the eigenvectors are in the U. So this is this matrix of eigenvectors that I showed many times before. And you rewrite it just with, like for each component, for each eigenvector and eigenvalue I. And you don't have to understand all of this, but believe me when I say that you can write this as a sum over all the entries in the connectivity matrix multiplied by the difference between all the different pairs. So it's the sum over the differences between neighbors. And again, neighbors is not neighboring brain areas as they are shown here, but it's neighbors in the graph as defined by the connectivity matrix. So in other words, the higher order the harmonic mode, the larger the eigenvalue. This is shown here. And the larger the difference between neighboring nodes are allowed to be. So, and that is exactly what leads to smaller chunks of the cortex being similar valued. So if brain regions that are connected in the graph are allowed to have very different values, the resulting gradient will appear less smooth and you have these smaller chunks of cortex. Okay, I will finish with a summary, which somehow has moved up here, but I will just tell you about it now. So what is so delightful about harmonic modes is that the underlying theory that I just vaguely touched upon links them to standing waves and self-organized pattern formation, like in the animal codes, yeah. They are eigenvectors and form an orthonormal basis, so they can be used to transform activity patterns into graph frequency space, just like a Fourier transform, but on graphs. So in other words, any activity pattern on the brain can be expressed as linear superposition of harmonic modes, and they are ordered by spatial or graph frequency, which provides a link to hierarchical network organization, and that makes them very interpretable. Yes, and I will end with a word of caution, which is that although these theoretical links exist, we still don't really know much about harmonic modes in the interpretation. And it's nice that they look like known networks, but that's not strictly enough to really know that the brain somehow uses harmonic modes as an organizational principle. Also, there are many techniques to related to what I showed here and many ways of computing the graph Laplacian or even just the connectivity. You can use different measures and we don't know which one is better or how to even how to measure what better or best means. So what I showed you, just as a quick information, is called Laplacian eigenmaps, but related techniques are, for example, UMAP, which I stole the example from, and there's also isomap and diffusion maps. In principle, harmonic modes and related techniques are just approaches to dimensionality reduction. And even though I hope that I have convinced you that they are rather more than that, the thing that you have to be really careful about is that you will always get a result when you're using this kind of analysis. And if you start with a structured graph that has some interesting properties, then you will always get a result that looks kind of nice. So you have to think about how you can validate results. Okay, so this is it from my side. I hope that you have many questions for me. And thank you for listening.